In this next video, we're going to talk about carbohydrates and lipids. And these are two very important molecules in order for life to be able to survive. So we'll look at these two different types of macromolecules in more detail in this video. And this is IB section 2.3. We'll cover most of the expectations for this section in the video, some of which we'll actually be doing in class. So to begin, to start out with our discussion here, we need to distinguish between what is organic and inorganic. And an organic compound is something that contains carbon. And these are found in living organisms. You look around, you see living things. They are made up of organic compounds that contain carbon, like a bird, for example. Inorganic compounds are those that don't contain carbon. There are some exceptions, as there usually is in biology, but an exception would include carbon dioxide, carbonates, hydrogen carbonates. Those would be some examples. An example of an inorganic compound would be like minerals that make up rocks. Now, as we look at or talk about these organic compounds that contain carbon, one of the most basic forms is something called a carbohydrate. And there's different types of carbohydrates. The smallest or the most basic is something called a monosaccharide. And that literally means mono is one or a single and saccharide is a sugar. So it is a single sugar. And these are generally used as sources for fuel. They can be converted to other types of molecules that we'll see here in a second, or can be combined together to make other molecules. Examples would include glucose, fructose, and galactose. And these can be shown or written a couple different ways. This is a representation of glucose, and these are both correct. It's just written or shown a little bit differently. Here as well, you can see them displayed a little bit differently. Now, what's unique or interesting about glucose, fructose, and galactose is they all have the same number. They're the same number of carbons, hydrogens, and oxygens. What makes them different is their arrangement of those. So these are all three examples of monosaccharides or single sugars. If two of these monosaccharides are put together, it can make something called a disaccharide, or two sugars. And these are also used as a source of fuel. They can also be converted to other molecules or combined into polymers. Some examples include maltose, lactose, and sucrose. And these are made or are different depending on what type of monosaccharides are put together. So for example, sucrose is a combination of glucose and fructose. Lactose, which is the sugar found in milk, is galactose and glucose, and maltose is made of two glucose molecules. The third and final type of carbohydrate is something called a polysaccharide, and poly means many, so it's many sugars. And these are made up of many monosaccharides linked together. And they are often used in plant cells for cell walls, energy storage, exoskeletons. Some examples would include starch, glycogen, and cellulose. You've probably heard of starch before, and that would be something that would be found in carbohydrates like potatoes or breads or pastas or things like that. Now you may be wondering, how do we get these di or polysaccharides? Well, that occurs through a process called condensation or hydrolysis, the splitting of them. And in condensation, for example, let's say we have two glucose molecules, and in order to put those two molecules together, something has to be removed. And what actually gets removed is water. And so this is called condensation. You could also think of it as dehydration synthesis. And so these two molecules are connected by the removal of a hydrogen and oxygen from one glucose and a hydrogen from a second glucose. And so these two molecules get linked together by an oxygen, and there's a byproduct of water. To split apart, to separate two glucose molecules or a maltose molecule, water has to be added. And so then what happens is we end with two glucose molecules. In lab, we will actually build these molecules, and you can see how they're put together and taken apart. But this is very important into building or breaking apart these polysaccharides. When you eat something, you need water in order for the polysaccharides, the carbohydrates that you're eating, to be able to be broken apart. Here's another way to look at it in kind of a summary of what we just talked about. Two or more monosaccharides or disaccharides join to form a polysaccharide. Polysaccharide can be broken into monosaccharides or disaccharides. That's kind of a basic introduction to carbohydrates, monosaccharides, disaccharides, and polysaccharides. In comparison to another molecule of energy is lipids or fats. And carbohydrates are often used as a form of short-term energy storage. They are soluble in water, as we talked about in our previous video. They can be transported around the organism pretty easily because they are soluble most times. They're very quickly digested, which is both good and bad because it releases lots of sugar if you need it, but if you don't need it, then that sugar just sits there and turns to fat. And it needs less oxygen to release energy. Lipids, on the other hand, fats, are longer-term energy storage. They're insoluble in water. They're not as easy to move around. They actually provide more energy per gram in comparison to carbohydrates, and it needs oxygen to release energy. Some similarities for the two: energy storage, and they're both organic compounds. In looking at some functions of lipids, energy storage, as we talked about, they are more efficient than carbohydrates because they have about double the amount of energy per gram of carbohydrates. They can be found in oils and plants and fish, fats and animals. They are grouped or stored in specialized cells called adipose tissue that's directly under the skin or around organs. And while some body fat is good in humans, having too much is a problem because that adipose tissue can start to affect the ability of those organs to actually function. Another thing that it's very useful for that you see in many Arctic organisms is blubber. It's insulation. It helps or is a way for those animals to stay warmer in the colder climate, and it provides protection against heat loss. And the last or third is buoyancy. Lipids are less dense in water, and so they actually help animals to float in water. So again, a lot of organisms that live in the Arctic or around the water, buoyant. Think of maybe sea lions. They have a lot of blubber that helps them to not only stay warm, but also. So then we need to get into and look at how are these lipid molecules actually made, or what are they composed of? And they are composed of something called a glycerol molecule and fatty acid tails. And it takes one glycerol molecule, right here, that connects to 3 different fatty acid tails. And the tails can either be saturated, monosaturated, or unsaturated. We'll look at those in a second. The saturated molecules have lots of hydrogen bonds and no double bonds. The unsaturated, less hydrogen bonds and more double bonds in it. And we'll take a look at those a little bit more in a second. Condensation and hydrolysis are also used to create these. So the glycerol molecule loses a hydroxide and an oxygen and hydrogen. The fatty acids lose a hydrogen. Water is removed. And so those 3 fatty acid tails are connected to a glyceride to form a triglyceride. And this is an insoluble molecule, as we previously talked about. Now, the difference between saturated, monosaturated, and polysaturated, this diagram here gives us a nice image of what this looks like. Here's the saturated, where the yellow circles or dots are representing hydrogens, the black is the carbon. A fatty acid tail is essentially a long string of carbons with hydrogens attached to it. The monosaturated, there's a double bond between at least a pair of the carbons, and so there's less hydrogens. Polyunsaturated has multiple double bonds, and so as a result there's less hydrogens. The hydrogen-carbon bond, the breaking of that provides a release of lots of energy. And so generally polyunsaturated and monounsaturated fats are considered a little bit healthier. Example would be like olive oil. Now, the arrangement of fatty acids can change a little bit, and this gets into a little bit of chemistry, but the positioning of hydrogens on a double bonded carbon, as we see here, the arrangement of these hydrogen elements, causes some changes called cis or trans. And so in the cis arrangement, the hydrogens are on the same side, if you will, of the carbons with a double bond, and trans, they are opposite. This is one thing that you will need to know and be able to recognize for the IB curriculum. Getting back to carbohydrates, as we've talked about lipids a little bit, some specific functions of carb functions in animals. Glucose, as we previously talked about, uses an energy source. It's a monosaccharide, it's small, it's easily absorbed, it's a quick energy source. Lactose, a disaccharide, is also an energy source, and it can be broken down to form a monosaccharide, and that's the type of sugar and a major component of what you find in milk used to nurse young. Glycogen is an insoluble energy storage in animals and fungi. It has a very branched shape. It's very compact. It's similar to starch in plants, but it's something that's found in animals, and it's primarily found in muscle and liver cells, and it's used for short-term energy storage. Starch, glycogen, cellulose are all of those are made by linking glucose molecules, yet the structures and functions are different due to the type of glucose used to make them in the linkage type between the glucose molecules. So basically what that means is that the linking of glucose molecules, or these monosaccharides, can occur on different positions within the carbon rings, and as a result that can cause these different types of molecules. And we'll take a look at this a little bit more detail when we start making models in class. On the opposite end, different functions for carbohydrates in plants. Cellulose is one of the primary ones, and speaking of the connections or the links between these different molecules, the link occurs between the first and fourth carbon between the different monosaccharides making up cellulose. And it's positioned next, the next glucose is positioned 180 degrees to the previous. And again, we'll look at this in the lab a little bit and get some hand-on time with being able to see this. And so it makes up a straight chain forming cellulose, and this can be used to form like microfibril bundles. It's something that's used in the cell wall and can help be used for protection and help that cell in the plant to protect it from bursting. Starch is another similar molecule, and its link occurs between the first and fourth carbon, but the two glucose molecules are orientated in the same way, and this actually causes a curved shape. Starch is used as a source of energy storage. It's often found in seeds to provide those budding or germinating seeds energy, and it's produced as an energy store. Some of the things that we didn't talk about specifically in this activity is examining and measuring BMI, body mass index, which we will do in class, and also some of the health risks and factors of lipids or fats. We will also do that in class as well as have a chance to build some of these different models.